So this is a series of videos I'm going to record on nutrients. And this first one, I'm talking about protein and amino acids. So the first thing is we hear about protein requirements. Is a protein requirement real? And the answer is actually no. There is no real requirement for protein. There is a requirement for individual amino acids. And so protein we need to eat as a source of those individual amino acids, of which 10 are absolutely required or indispensable, and the remainder are dispensable. So when we use protein requirement and we use this to set the approximate protein level in a diet for a certain species of fish, what we're really doing is using this protein requirement as a guideline for approximating the right quantity of amino acids the animal needs to eat to grow based on the amount of expected feed consumption. So this is very, very basic, but we have protein structure here. And so we start out with a complex protein. And then if we consider how this protein is broken down, we see each one of these 4 subunits. And each one of these subunits is made of a helix of proteins. Protein which is based on a certain combination of amino acids. This is the secondary structure. And the primary structure is just simply a row of amino acids strung one after another in a certain sequence, and this is genetically controlled by the DNA and by the mRNA synthesis and so on. Now, on the other hand, when we take this intact protein in and we digest it first of all in the stomach and then in the intestine, we have proteolytic enzymes, pepsin, trypsin, chymotrypsin. And they break down the intact protein into peptides. These peptides could be T2, 3, 4, 5, or more amino acids in a small group chain. And then we have aminopeptidases. So these could be an exapeptidase, meaning that it will cut a peptide from an end and go in to the other end. Or it can be an endopeptidase where it will target an amino acid in the middle of a peptide and start breaking it up amino acid by amino acid from there. So this is just showing, once again, very basic, the whole range of amino acids there are and their general structures. This shows you whether they have a polar charge or not. And we talk about hydrophobic side chains. These means these are repellent to water. We have uncharged, so they're kind of neutral. And then we have charged side chains up here. Positive in this case and negative over here. And then if we look at this paper, Amino Acids, Metabolism, Function, and Nutrition, and you can scan this one and download it from there, we see that he has compared the essential and non-essential amino acids for mammals and fish over here and for poultry. And you can see that there's a slight difference down here. There's a few that are different. But in general, most animals have the same essential amino acids. So as I said, we talk about a protein requirement and we describe it as a percent of the diet. This is the traditional way of talking about protein. We buy a bag of feed, it says minimum 35% protein. So a dietary protein requirement expressed as percent protein in the diet without considering protein digestibility or protein quality really has limited usefulness. Any undamaged protein And undamaged is important to understand. Undamaged protein coming from any plant or animal source that can provide digestible amino acids can be used in feed to supply dietary amino acids. The liver does not care where the amino acids came from. As long as the food source can be digested in part or in total, then it can supply those digestible amino acids. It is good for feed. So we have this table from NRC 2011 with some recommended dietary protein levels. So we have here Atlantic salmon, so that's a carnivore. Channel catfish is omnivore. Common carp is herbivore. Nile tilapia, well, carnivore, and a carnivore, omnivore, herbivore. And then we have Pacific salmon, rainbow trout, sea bass, and gilted sea bream. These are all carnivores. And one of the things that we notice is in general, carnivores say 48% protein for very small fish. And we see small fish generally have higher protein requirements than we see large animals over here. And it's really at the small size, all animals are essentially more or less carnivorous because they're growing very fast. They need very high levels of protein, and they get that best from eating other animals. But as the animals grow and they begin then to move towards their preferred diet, for example, we look at the salmon here, or we look at the herbivores, we can see that the protein requirement of these large herbivores is considerably less than for the carnivore. Now, when we start talking about protein sources or protein quality, we have to consider that different species, due to digestive system differences, that is in terms of their gut length, their physiology, their digestive enzymes, are assumed to tolerate different sources and of feed and the quality of the protein in the diet. And this is a very famous graph. You can find it everywhere on the internet. It came from Dr. Linwood Smith from the University of Washington. This was a course that I was involved in teaching. And what he's done here is he's shown you that whether we're talking about a trout, it has a distendable stomach and a very short, straight gut, or whether we're talking about a herbivorous fish like a carp, or we're talking about a fish like the milkfish, um, then we see that there are substantial differences, especially in gut length. And sometimes we have, in the case of trout and some other fish, we have something that's pyloric. This is an enzyme secreting tissue versus other fish that don't have such things. And I give you this example. So many carnivorous species don't have the enzymes needed to digest plant-sourced ingredients. So when we start talking about where's our protein going to come from, well, then we have to consider, well, is it embedded in a lot of plant um, cellulose, or is it embedded into plant cell walls? Because that then becomes a difficulty, because those carnivorous species don't have amylases, xylanases, they don't have mannanases, and they don't have glucanases. So when we start talking about these complex polysaccharides, they can have inhibit digestion, inhibit nutrient retention, and the carnivorous species simply do not have the enzymes to break them down, which then, due to cage effects or to, let's say, gelling effects, can actually capture valuable nutrients, and they will all be excreted. So these are unable to be utilized by the fish. So we generally will see when we feed a carnivorous fish these diets with a lot of plant proteins that are embedded with a lot of polysaccharides, that the available digestibility of the protein and amino acids tends to be low. So here if we look at protein intake, now this is protein intake per kilogram of live weight gain in different fish and shrimp. And then we also have pigs here, and we also have chickens. So what does this tell us? Well, if we look across at all of these fish, we see that there are some differences. If we have a low value here, this tells us that the grams protein weight gain, or protein intake versus weight gain, lower is better. It means that we have less intake per unit of gain. Now, when we have a higher intake needed to get a certain amount of gain, this suggests that the digestibility, digestion efficiency, the absorption efficiency is going to be somewhat less. And this is going to be one of the things, as we mentioned, that's going to differ from species to species. Now, talking about protein intake. So when growth is fast, relatively more protein needs to be consumed, and when growth is slow, less protein intake is needed. And we often will um, estimate that, for example, a feeding table will estimate that an animal needs to eat this X amount of food because we're calculating how much protein, how much fat it's going to be taking in on a daily basis. But rather than talking about feed in terms of percent of protein or percent of the feed, I prefer to look more directly at this idea that we measure protein intake in terms of grams of protein in grams of weight gain per day. Now, this graphic is showing us the whole body chemical composition of rainbow trout obtained from 66 studies conducted between 1976 and 2005. And once again, scan this and you can go and find it. And so what we can see is that across a wide range of body weights, the rainbow trout, we see that whether we're looking at water or moisture content of the flesh, whether we're talking at dry matter or protein or lipid or ash, uh, the quantities are quite consistent. So if we're talking about how much protein in a gram of weight gain per day, we could look at this and then we can say, well, it's about like that. And we could just kind of go back here and say it's a little under 20%. So if a fish gained 1 gram of weight per day, we could estimate that probably around 18 to 0.18 to zero point 0.2 grams in that 1 gram would be protein gain. Now, we've seen already in that previous table that there are differences in our recommended protein levels, and this really depends on the adaptation of the animal to its natural food sources, whether it's a herbivorous fish, an omnivorous fish, or a carnivorous fish. And in this table, we can see that we have at the top a bunch of omnivores. Now, I wouldn't necessarily consider an Indian major carp an omnivore. I'd consider it, and the common carp, these are more classified as herbivores. And as an adult, milkfish is also basically a herbivore. Channel catfish, silver perch, as adults, they're basically omnivores. And then we have a bunch of marine species down here. These are all highly carnivorous fish, as we can see. So we have a diet, and our diet has 100 units in it, 100% in it. And then we look at it and we say that we need to have, for sea bass, that we need to have 43 to 52 out of those 100 units in the feed have to be composed of protein. But in a fish like a tilapia, only 26 to 35 units. So what's the difference between 43 to 52 and 28 to 35? In most cases, when we're formulating feeds, this difference is largely due to the presence of carbohydrates, of starches. And so that takes up some parts, 15 or 20, 25 parts of our 100 parts. And so I suggest to you that part of the reason we have these different protein levels depends on how a fish is able to tolerate carbohydrates. And so this is partly why they have a preference for high dietary protein feed levels. And so we see here that herbivores and omnivores that tolerate digestible carbs or dietary carbs as a source of energy, whereas carnivores do not tolerate carbs as a source of energy. The carbohydrate content of the feed is low, and that space is filled up by simply adding more protein. So we know that there's a lot of things which can affect the requirements for dietary intake. And so here I'm looking at this research on a lysine requirement of tilapia as an example. So do requirements change with fish size or not? So what are we looking at here? We're looking at body weight. We're looking at lysine requirement as a percent of the feed. And notice it really does not differ. 1.4, 1.4, 1.4. It does not really differ. And if we look at it as a percent of protein, once again, we don't see a lot of difference. Now, this paper is available, but I wasn't able to find a place where you would be able to download it for free. So this takes you to the publisher's website. Now, the questions when we look at this table, what about the impact of feed intake on the requirement? And what about the impact of feed protein amino acid digestibility on the requirement? Well, when we're talking about as a percent of feed, we're not really able to know what else is in the feed that may or may not impede lysine absorption. And we also don't really know whether those ingredients might have had some of their lysine damaged through improper processing, maybe excess heating and so on. When we look at lysine as a percent of protein, this in general is preferable because then as our feed level changes, the amount of lysine will increase correspondingly to the amount of protein in the feed. So this one is like automatically compensating, where this one as a percent of the feed is really not doing so. Okay, so just now we saw if we report lysine as a percent of feed or as a percent of protein, that it isn't really different. But if we start looking at lysine requirements expressed as an intake as a body weight and not as a percent of feed, then what we see is that in our table here, we have initial body weight is 20 grams. And what we see is the requirement is 11.9 milligrams of D-lysine per kilogram body weight per day. But when we look at a larger fish, 165 grams, the amount required is 58.7 grams digestible lysine. And so that shows you that if we consider it on a available lysine per body weight on a daily basis, that a large animal simply is growing more body mass, not as a percent of body, but as a just simply as a gravimetric weight gain. And therefore, it's depositing more protein. It needs to eat more lysine. Now, the final say on determining protein requirements, and this is a quotation from nutrient requirements of fish and shrimp. I'll just quote it. Estimates of protein requirements should be considered highly approximate, especially in the current context in which an increasingly wide variety of feedstuffs is used in the formulation for feeds for different fish and shrimp species. So this tells us that we have to consider the availability of the protein in those different ingredients. And so that might involve digestibility studies or something like that. But whenever we see protein requirements listed, just keep in mind that they're only guidelines and that you can have um, a low quality protein at a high level and it won't do very well. Or you can have a lower level of protein, but a very high quality protein, and you will actually maybe obtain better growth. Okay, I think this will come up a bit later. I have a slide here somewhere. Okay, so when we start talking about amino acids, after all, the protein is there only as a source of amino acids. The first question is whether an amino acid is essential, also known as indispensable, or whether that amino acid is non-essential, also known as dispensable. And with amino acids potentially being used for either protein deposition or catabolism for energy, sometimes indispensable amino acid requirements are difficult to estimate without being very careful about our experimental designs. There are many complicated factors which can impact our estimation. So this is a study, very old, from Halver and Hanks from 1960. It's with sockeye salmon. And what we need to do is determine which ones are essential and which ones are non-essential. And this is a form of Halver's H440 based test diet. But in this diet, we're not using intact protein like casein or gelatin. We're using individual amino acids. And so this gives us the ability to delete any one out of this total. For example, we could make a diet with 0 methionine in it, but every other one is showing the level as you see it here. And then we could add in a little bit and a little bit more at different levels, and then we'll see what happens. So when we delete out of this and we start to feed the fish, what we see is that when it's totally deleted, no growth occurs. Whereas if it's all there, the growth occurs like this. Now what they've done here is they've let the fish grow to about half of the rearing time, and then they've switched them from a 0 to a diet containing the amino acid, and then growth begins to recover. So this is how we know that if it's not there, no growth will happen. If it's there, growth will happen. So that makes it essential. And then what we see here is that it was added, but at a much lower level. And so we saw a little bit of growth, but not as good as if it was there at the full quantity required. On the other hand, when we feed a non-essential amino acid, it doesn't matter whether it's there or not there, growth will occur unimpeded. Now, there are many ways to determine quantitative amino acid requirements. And in the old days, before we had a lot of sophisticated laboratory equipment, we were really relying only on things like growth response assays, whole body amino acid profile, but now with molecular biology, we can then do amino acid enzyme studies, for example, and we can look at enzymes and we can look at oxidation studies. And then we also now see that we can have factorial models which estimates maintenance requirements, rates for growth, rates for faster growth, and these factorial models, which I believe are a very excellent way to do this kind of research, can be done at different temperatures and different sizes, and then they really give us excellent data to be able to estimate a quantitative requirement for any environmental condition and any body size. Now, this is a paper where this author is looking at interspecies differences in arginine and lysine requirement, and he's covering a bunch of different animals, but we have rainbow trout here. So what we see here, and this is basically extracted right from the paper under this graph. So we see the metabolic responses to dietary arginine intake relative to changes in protein synthesis. So protein synthesis is this line right here. Now we know that there are a number of other processes that occur simultaneously as part of the process of producing protein. For example, it could be like genetic expression, maybe like myosin production or something else. And so we could look at these 3 points and we could say, well, this one we can measure, but maybe if we measure this one too and this one too, then we'll have 3 ways to measure the response of the animal to arginine intake. And they give the example here, we could look at nitric oxide or urea or creatine synthesis, and we can then say that looking at several of these will give us more confidence that our estimation is correct. Now, one of the things, though, is that there are other possible responses to arginine intake that we could potentially use that are looking at different metabolic functions. And in this case, arginine intake required for the maximum response can be different from the requirement necessary for maximum protein synthesis. And so what we're showing here is that there are several other lines here with the blue circles. And somebody might measure some of those, but are they going to be giving us the confident requirement or a confidence in some published requirement that would be acceptable to us? And I would have to say, well, if we stick with these red circles, we're more likely to get a reliable estimation. And I have another graph right here to show you. This is from a paper from quite some time ago, from 2000. And it's just showing you that these different methods of estimating a threonine requirement. So here's our original weight gain data. We just simply measure the weight of our fish and we plot the weight gain over time on the different diets. And so then we come up with this simple thing. Now, we can take that and we can plot it as a broken line model. And so the black dots here representing the same data set as this. And we can then use a broken line model. Now, broken line model were very popular in the 50s and 60s and maybe extending into the 70s. But as we started to come more towards modern methods of analysis, we began to look more at things like quadratic equations and multinomial, polynomial equations. Because if we look at this one, we can say, well, there seems to be an inflection point right there. If we plot this as a line, we can use analysis of the points to say there's a line here, and obviously there's a line here, and we can say, well, that's the requirement. But notice that when we use polynomial instead, we end up with a value slightly higher. Sometimes the paper will report it as the range is between this value and this value. They won't give you one number. They'll say, let's say it's between 3.1 and 3.3, something like that. And then they're kind of hedging their estimation. And there's really nothing wrong with that. I think in many ways that's a more honest way to state it than just giving you one number. Now, one of the complications with aquatic animals is they are very susceptible to using protein for energy. And that's because most aquatic animals, while in water, favor energy production for protein, and this excludes sharks, terrestrial crustaceans, and walking fish while out of water. So walking fish, what's that? Well, walking catfish, snakehead, anabas, the climbing perch, and then of course at the seaside, maybe you've seen gobies, living in the mud on the seaside, and they climb out of the holes. While they're out of the water, they are producing urea, and when they're in the water, they can be just excreting ammonia. So I made some notes here. So we see the release of protein-derived nitrogen as ammonia directly from the blood through their gills into the surrounding water. And that is favored because it has a very low metabolic cost and no storage requirement. Very simple, very cheap for the metabolic process of the fish. And it's only impeded by excess dissolved nitrogen in water, reducing the concentration gradient across the gills. So this is why we need to be concerned about dissolved nitrate, dissolved nitrite, and dissolved ammonia. So terrestrial animals, on the other hand, um, and that would include our terrestrial crustaceans and our walking fish while they're out of the water, they have to sequester toxic ammonia in non-toxic forms. And so they produce either urea or uric acid, and it's excreted with the urine. And because the nitrogen has to be captured, it has to be produced into a non-toxic form, it has to be stored in the blood in a non-toxic form, it has to be excreted as urine. This has a high metabolic cost, but there is no alternative. So fish can do both, or but land animals cannot. So when we consider then again energy, we see that different species, depending on their habitat and normal dietary intake, have different preferences and capabilities when it comes to producing energy from nutrients. And so we've kind of hinted at this previously. So if we look at protein catabolism for energy, and this graphic is a little bit small, but what it shows us is that our food comes in, we have proteins, carbs, and fats. The proteins are converted into peptides and into individual amino acids to free amino acids, and then they are absorbed into the citric acid cycle, the Krebs cycle, and the ammonia is excreted as a byproduct. So we see here proteases reduce the protein to peptides and free amino acids. Amino acids are transdeaminated, and then the nitrogen is removed. When we're talking about alpha keto acids, we're really talking about um, pyruvate and maybe in some cases acetate. So on the other hand, the fatty acids go through a different process, and the carbs go through a different process. And if you want to know more about that, I recommend looking for this paper, a broader look at ammonia production, excretion, and transport in fish, a review of impacts of feeding and the environment. And so this one, I believe, is also not free, but this is the publisher link. You can go there and find it. So I mentioned earlier protein quality. So when amino acids are not balanced, relatively more protein needs to be consumed to provide the essential amino acid requirement, because temporary excesses of free amino acids will be catabolized. And I've talked about this in another video where we're looking at what we would call, an essential amino acid. Now, sometimes the nutritional quality of a protein is estimated by the amino acid index. And this compares how closely the amino acid profile matches a high quality reference protein. So what would be a high quality reference protein? Someone might use it for egg protein. Someone might use it for freeze-dried cod protein or muscle protein of some fish species or another. And then we would then compare our feed ingredient. It could be a plant ingredient, it could be an animal byproduct. And then when we compare them using this amino acid index, it will tell us, whether it has a higher ranking or a lower ranking. And in general, a very high quality protein like fish muscle protein or egg protein or so on tends to have an extremely high number, and almost any feed ingredient will have a lower one. Now, often we can blend different ingredients to balance the amino acids that we want. And this has been the traditional way of doing that. We look at each ingredient and its composition, and then we try to combine them in such a way that we meet our requirement. And so this has been the standard we do with shrimp, because until recently, we were not very confident about adding crystalline amino acids to shrimp feed because they're water soluble, they would leach out. And so we resolved this problem by really focusing on blending ingredients. However, when we're talking about fish which eat their feed rapidly, and we don't really need to worry much about leaching, we can use crystalline amino acids, but they have to be used carefully because they can be absorbed very rapidly before the amino acids from the ingredients are digested and become available. Could be minutes later. The consequence of that is whenever you have these instantaneous imbalances, you often then times have amino acids being shunted off for energy production. Now, we've talked about the differences in the physiology and the morphology of these different organisms. The graph that I showed you, that each of the different types of fish has different stomachs, different gut lengths, different enzyme secretions, and so on. So these need to be considered when we're talking about protein intake and our feed quality. But in general, to meet the protein requirement necessary for unimpeded daily weight gain, that is maximum growth potential, we can feed a lower protein feed fed more often, and that can provide the same amount of protein in grams per unit per day as a high protein feed less often. Now, there's a caveat here, and that is that a fish with a very small stomach cannot eat a huge amount at any one time. It turns out to be more like a grazing animal, like a tilapia. We could call more or less like grazing, like similar to a cow grazing. Whereas a fish like a catfish or a trout or a salmon, these fish are often not eating every single day in the environment where they live, and they have a stomach which can be expanded, and they can swallow an entire prey animal all at once, and maybe they don't eat every day. And so in that situation, the size of the stomach and the extendability of the stomach will have some impact about, and also the gut passage time can have an impact on how many meals a day an animal can actually imbibe. So then finally, we can have a lower protein feed made with highly digestible protein. It provides the same amount of protein on a weight basis as a high protein feed made with poorly digestible protein, because in the end, we're only concerned about digestible amino acids coming from highly digestible protein. Now, looking at the way that in research, that we assess protein retention and utilization, as we understand, protein is consumed to provide amino acids that may be converted and incorporated into new tissues for growth and bioactive proteins like hormones and enzymes, or those amino acids may be catabolized for energy, and in that happens, they're lost to growth. So we have the protein efficiency ratio, PER. Now this is quite an approximate estimation because it's based on wet weight gain, and we're measuring the protein maybe using a method like approximate analysis of the fish body tissue. And then we're calculating the protein intake from the feed. Now, this we need to know then the amount of protein at the beginning of the experiment inside the fish, and the amount of protein that we measure inside the fish in the whole body of the fish at the end, and we take the difference as the gain. And then we take the protein intake from the feed, because we make a feed and we analyze the protein content. So that's how that's done, but it's kind of like an approximation. We can also measure protein retention efficiency. So this one then is measuring protein gain, more or less measured in the same way as with the PER. And then we have protein intake from the feed as normal, and this one we then calculated as a percent. And another method called net protein utilization. So there's the equation of how you calculate it. So we have four factors: B, the total body nitrogen of animals fed the test diet; the total nitrogen BK, the total nitrogen body nitrogen of animals fed a protein-free diet. Now, with fish, feeding a protein-free diet is extremely difficult. The feed is very, very poorly accepted, and in general, because it does not provide any protein, fish are very sensitive to that, and these kind of experiments oftentimes will result in very high mortality. Nevertheless, then we have another factor, LK, which is nitrogen intake of animals fed the protein-free diet and nitrogen intake of animals fed the test diet. And so we estimate the differences and we put them into this equation. So to summarize, when we talk about a protein requirement, we are using that number as a rough guide to the quantity of amino acids an animal needs to consume on a daily basis. We can say, well, in general, protein is nitrogen times 6.25. That's based on the generalization that protein contains 16% nitrogen. However, many ingredients have different ratios than 6.25. Soy is maybe 5.7, 5.4, somewhere in there. Okay, so protein deposition is the basis for all weight gain, and for that to happen, those 10 essential amino acids have to be obtained from the diet, and they have to be all present all of the time. A deficiency in temporarily or not of any one of those will result in a reduction of growth. And then protein intake and amino acid intake varies with the quality of the feed and its ingredients. And everything we need to consider is on a digestible nutrient basis, and that is impacted by the processing of ingredients, the amount of heating, the amount of drying, and then also our methods of feed production, whether we're producing by pelleting or extrusion or whatever means, because that also then has temperature and other factors that can impact the digestibility of the protein and the resulting supply of amino acids. So to finish off, I want to give you some recommended references, and this is to help you to understand this topic more if you want. Now, these two papers here are a pair published by the same people in the same year, 2003. And here's the QR codes. You can download these for free. So you have this one where they're looking at the essential amino acid pattern of rainbow trout to maximize nitrogen retention, or in other words, growth, and minimize nitrogen excretion, or otherwise minimizing waste. And then the second one is looking at the ratio of the essential amino acids as determined in this experiment. And here then what they do is they put essential amino acids to a variety of balancing a bunch of different ratios of non-essential amino acids. And then they look which one, once again, maximizes nitrogen retention and minimizes nitrogen excretion. So this then is a kind of a way to determine which of the non-essential amino acids make a greater contribution to growth than maybe some of the others. Then I have this other paper, protein and arginine requirements for maintenance and nitrogen gain in 4 teleosts. So as a technique, as a way to look at it, so they have 3 diets: high, medium, and low protein diets. And they were fed to 4 species: rainbow trout, turbot, European sea bass, and gilt-head sea bream. And each one of those low, medium, and high protein diets had different levels of arginine. So they were fed either 1, which was extremely deficient, 22, or 29 grams per kilogram of diet. And that was based on an estimation that the requirement was actually around 15 grams. So you can go in there, they have lots and lots and lots of graphs, and you can see that the importance of the protein, is the protein level important, or is the amount of the amino acid maybe more important? A low protein with a essential amino acid like arginine, maybe the combination of low protein and high arginine can perform nearly as well as high protein and high arginine. So they investigate all of that. Then looking at shrimp, we have current status of amino acid requirement research in marine penaeid shrimp. So this one is 2006. This is kind of like a review paper in a way. And it also looks at techniques and research methods, which I think is a valuable thing to read and learn about. And then we have essential amino acid requirements of fish and crustaceans, and this is a meta-analysis. So a meta-analysis is when they take tens or dozens or maybe even hundreds of different studies that have looked at different requirements of individual amino acids for different species. And then they kind of try to put it all together and pull a idea out for what are the core essential amino acid requirements. And a meta-analysis kind of helps in a way because it kind of de-emphasizes the laboratory techniques, the laboratory rearing conditions, and it tends to focus more on the results. But of course, each one of those is affected by the way the experiment was conducted. And so they will also go in there and they will discard methods, research papers, and so on that do not meet a minimum standard. And then finally, I have here the dietary protein requirement of Pacific white shrimp in 3 different growth stages. And so what I find interesting about this is that if we talk about different growth stages, we're talking about different requirement on terms of daily intake per weight gain per day. And so this is a good paper to look at to understand this concept better. And this is paper from 2018, and you can find it and download it from there. So that's the end of this presentation. It's a little bit longer than some of the other ones that I've done, but I wanted to do it with this full set of slides because some of these points then I won't have to talk about later. And here I have a picture of some experiments I was doing with red tilapia. This was a Taiwanese strain. They're a little bit different body shape than some of the other red tilapia that are out there. And once again, to conclude, you can find me on YouTube here. You can find me on X, and you can find the audio transcripts of these presentations on Apple Podcasts. Thank you very much for paying attention to my video, and I look forward to recording more for you.